Double wired and ready to go. We can get underway. I'd like to.
I'd like to welcome you all to our grand rounds today. As you might notice, the person to my left is not Judy Rice. So there's been a change in the schedule. As many of you may know, she was scheduled to talk to us today about Johnson City Community Health Center and treating the uninsured mentally ill. But she canceled because she's ill with pneumonia, so we're looking toward rescheduling her once she's better, and we hope that will be soon. We're very fortunate to have been able to garner the interest of Dr. John Hendrick to step into her place on short notice. And while he needs no introduction for many of you, I did want to take a few minutes to introduce him. Dr. Hendrick is Chief of Psychiatry at Mountain Home VA. He's an Associate Professor of Psychiatry at East Tennessee State University and directs the Neuroscience Didactics track for the Psychiatry Residency Training Program. A graduate of the Medical College of Virginia, where he also trained in psychiatry, he's a Distinguished Fellow of the American Psychiatric Association. He holds a Distinguished Service Award from the United States Public Health Service, has been named an Irma J. Bland Awardee for resident teaching by the American Psychiatric Association, and has been twice named Resident Teacher of the Year at ETSU. His focus is on behavioral neurology and neuroanatomy as it applies to behavior, and he's going to present an amalgam of information from several of his talks to us today in an attempt to better understand the functional complexity of our individually personal 3-pound universe known as the brain. And so without further ado, let me turn things over to John, who will address us today on advancing the concepts of behavioral neurology. John.
Thank you, Dr. Dick. Well, whenever we're going to do a talk as biologically oriented as this one tends to be, I always like to remind everybody that there is a psychodynamic reason that we're interested in all these things. So I start with Dr. Jung and his observations, and we will soon move to Dr. Freud, because all of these things are significant in understanding brain and behavior. So, thank you for the introduction, Dr. Goodkin. This is my disclaimer. I have no disclaimers other than I work at Mountain Home and at ETSU, and that's what I do. Now, there may be a video slide at the end that does have a, I think it's actually weirdly a Dremel advertisement in it. If it downloads and I play it, it may come up. So it's not an endorsement on my part. The good news is probably the video won't play, so it may not matter. So, behavioral neurology. Here's a true story. An attorney cross-examining a pathologist in a death case offers him a moment of testimony to which you should all aspire and do as well. The attorney says to the pathologist sworn on well, I'm a pathologist and I specialize in death cases. So the attorney says, well, yes, but when I go to my doctor, he does a certain set of things, and I wonder if you did those with the victim. And the pathologist says, I'm not sure what you're talking about. And the attorney says, well, every time I go to my doctor, he measures my vital signs. Did you take this man's vital signs? And the pathologist said, no. And so the attorney said, well, then how were you for sure aware that he was dead? And his answer was rather telling. That's why. So meanwhile, that's a cute story, but it doesn't quite end there. The pathologist, seeing his opportunity, stated, now that I've had a moment to reflect on it, I suppose he could be out there somewhere practicing law. So meanwhile, whether we know that man was dead or not, it's certainly an example of the fact that the issue in behavioral neurology that we're all fascinated by, psychodynamics, nature of consciousness, how does consciousness play into brain and behavior? So this issue of consciousness is very interesting. About 13.7 billion years ago, this Big Bang occurred, and things then happened. Around half a billion years ago, Cambrian explosion occurred, and life had existed prior to the Cambrian explosion. But at the time of the Cambrian explosion, life forms became multiply diffuse, many different life forms. However, they were very simple, creepy crawly kinds of life forms. But they did have these sensory motor loops that are so important in understanding the nature of consciousness. So they could have rough visual systems and sensory perceptions other than vision. But then it was not until about 1 million years ago that a being named Homo erectus came available. And Homo erectus had a pretty respectable brain. Homo erectus habited this earth much longer than Homo sapiens have, but nonetheless, eventually didn't have enough capacity to adapt consciousness to be able to survive environmental changes. Neanderthals followed, and Neanderthals, probably somewhere in the 300,000 to 250,000 years ago range, are actually still evident in human DNA today. We can still see them there. So there was an interbreeding, and at about 250,000 years ago, Homo sapiens emerged. But it was not until roughly 50,000 years ago that Homo sapiens moved into the nature of what we now know as modern man, or Homo sapiens sapiens. The main issue here, the reason we use the term sapiens, after all, is to understand things effectively. Reason we double the term here is because, as far as we know, man is the only creature capable of the amount of verbalization that it has, using that verbalization for pedagogy or teaching its young, using that as a survival device, and making progress with tools never seen elsewhere in the animal kingdom. So how did we come to be this way? Well, first of all, we got bigger brains. So when you look at lesser primates, you see smaller brains. But chimps really don't have that much less size of brain, given their body volume overall, than humans do. And you'll notice a couple of things. There's some specialization features here. First, look at that cerebellum on chimps, larger per capita than it is in humans. Well, they have better balance, better motor skills in many ways. But humans, on the other hand, have their remarkable left brain. The left brain, especially the arcuate fasciculus, allows for a level of vocalization that becomes distinctive verbalization that is nowhere else seen in the animal kingdom. And that is one of the key things separating man from other primates. However, obviously the volume of the brain is part of that as well. But we see here, really we're not the largest brained animals. Dolphins actually have a larger brain, so do elephants. Elephants, I think, on the basis of size per se, though elephants are very interesting because they share with us remarkable memory capacity and mirror neurons, much more so than dolphins do, apparently to us. Now, dolphins, you'll notice in these past two slides, I want you to notice as primates go up the scale, there's increased gyration. Lysencephaly becomes increasingly sophisticated as you go up the phylogenetic tree. And the lysencephaly here is also very interesting because if you look at the gyrations in sulci and the dolphin, you'll notice that compared to the human, where there's left-right differentiation, in dolphins, the brain is very symmetrical. Well, why would that be? And from an evolutionary point of view, if you think about it for a minute, for a dolphin, attack from left or right, attack from above or below, attack from in front or behind is the same danger risk. So from an evolutionary point of view, dolphins have to be very symmetrical in their coordinated movements, whereas humans do very well to separate the sides of the brain for various and sundry reasons. The right brain to watch the environment carefully, while the left brain controls the majority of coordinated movement and also verbal skill. Well, those are interesting ideas, but of course, in consciousness, the real question becomes, why do we have such a tremendous advantage? Well, one of the big reasons is the planning we're able to do from our frontal lobes. Now, the diencephalon and its resident limbic system is not particularly larger as you go up. It's just kind of larger depending on the size of the brain. But the frontal lobe, the telencephalon in particular, is huge in humans compared to its relative peers. But even that isn't enough to explain the nature of what consciousness really must be. In fact, it is the level 1 association to association neuronal connections in the cortex that cause consciousness, that cause the remarkable skills that humans hold that lesser animals don't. So, in the very outer layer of the cortex, in the gray matter, those associational neurons are tremendously more connected than in other species. And they allow, not only in the frontal lobes, but also in the other portions of the cerebral cortex, most notably, though, in the frontal lobes, they allow a tremendous amount of associational thinking that eventually allows us to have the full degree of human consciousness that we experience. So, how does this make a difference? Mammals in general are very capable of learning. Okay? Once we get to primates, social orders become very important, and they have a working computational model. They pay attention to hierarchies in their groups. That's very important to their evolutionary niche. Once we get to great apes, there's theory of mind. And for those of you that are unfamiliar with it, this is a concept in which you realize that your behavioral actions interact with those around you, and that their behavioral actions in return interact with you in such a way that you begin making associational derivatives, decisions about how to behave, and you make decisions about your own behavior in context of the behavior of those around you. Now, the great apes are capable of that, and they're capable of vocalization. They can certainly scream at you, but they can't talk to you. And that's where mankind's huge jump curves. So in this Darwinian concept of evolution, along comes Freud. And Freud says, there's more to it than this. There's an unconscious mind. There's a preconscious area of the mind. And the mind then has a small portion, but very substantial, I think we would agree, that is conscious. Now, Freud alone came up with this idea, and I think we have to give him tremendous credit because all theories of consciousness, basically, are very integrated with this tripartite model one way or another. It also seems to be a basic underlying structural phenomenon for brain itself. Brain often has tripartite organization. But even with this idea, Freud became dissatisfied and later needed to move to a more structural theory in which the relatively unconscious id pushed drives forwards into the reality-principled ego, and the ego was controlled by the conscience, or the superego, which had both conscious and unconscious portions. So, Freud came up with this excellent theory. But what he spent much of his career stymied by was the fact, given the technology of the time, the biology of the time, he didn't know where these things were, and he always wanted to know. So we kind of do now. Here is the anterior cingulate cortex. And I digress just a minute to say that Dr. Lloyd kindly had a slide for us last week that said midline brain structures. And again, residents in the room, midline brain structures isn't enough. It's the anterior cingulate cortex, particularly the subgenual gyrus. Okay, so this portion right down here that are very important in pushing motive drive forwards in the brain. Now, there's a bidirectional connection between the anterior cingulate cortex and the frontal lobes. And in addition to that, not seen here are the basal ganglia. You can imagine where the basal ganglia are, and you'll see them in a later slide. But you can imagine them superimposed in the diencephalon region here. The head of the caudate has very special relations to the frontal lobe. The head of the caudate nucleus has bidirectional connections with the frontal lobe, as the anterior cingulate cortex does. In general, though, the basal ganglia have descending motoric influence from the cortex, not so much this bidirectional feedback loop. So here comes motive into the frontal lobe. The frontal lobe also has an interesting connection with the caudate, head of the caudate nucleus that doesn't otherwise exist between cortex and basal ganglia, and there's a reason for it. The frontal lobes themselves, especially Brodmann's areas 9 and 10, represent executive function reasons. So motive drive, what Freud talked about as id, is pressing into the frontal executive regions. Executive functions are occurring at this level. Processing then is being fed back to the anterior cingulate cortex, but also to the head of the caudate. And this information then is the kind of thing that we work on in reality, or the kind of thing that Freud would have referred to as ego. And then the orbitofrontal cortex turns out to be the site of superego functions, and we largely know this because of a variety of reasons. We see lesions in this area, tumors or trauma. We have evidence from prefrontal lobotomies, what the effects are here. We see the superego functions are basically located in the frontal cortex. So much of the nature of consciousness and its associated psychodynamic world have a lot to do with the organization of the frontal lobes. And they look kind of like this. Basically, they are motoric in a way. The motor strip is in the frontal lobe, but there is a premotor planning area that is very influential in utilizing this motive information, motive information, to drive it into the motor cortex before motor decisions are made. And you're going to be surprised how much information drives that way. Then there is the prefrontal region, which is responsible for largely those executive functions that I just mentioned. And then in green here in the midline, you see the anterior cingulate cortex and its very influential position. The anterior cingulate cortex is essentially the modest cortical remnant of the diencephalon, or the cortex of the limbic system, if you will. Now, the frontal lobe then, first of all, here you can see its close connection with a poor battery. You see its close connection with the anterior cingulate cortex. And we see here the dorsolateral prefrontal cortex, lateral orbital, and medial orbital frontal cortices. Now, we'll start with those for a minute. Those three represent the bulk of executive function areas. Dorsolateral prefrontal cortex is very much involved with executive decision-making and planning. It's probably quite abnormal, especially in its coherent output in schizophrenia. And then the medial and lateral orbital prefrontal cortices, for purposes of this discussion, are largely involved with valuation. So in other words, either valuing emotions or valuing things, putting value on them and deciding what that means. Now, having talked about that, you'll also see a couple of influential regions here. The premotor cortex and the motor cortex. And then two other regions that I want to talk about just briefly, the frontal eye fields and the supplemental eye fields. Again, supplemental eye fields are planning for activity. Frontal eye fields are helping the occipital cortex put the eyes in proper position to determine what's going on in the real world. This is very important. Interestingly, they run a track through the cingulate gyrus, which we normally think of as part of the limbic system, but there's a tract from these frontal eye fields into the occipital lobe through the cingulate gyrus that has much to do with visual processing and getting the eyes in proper position for determining the nature of the reality of the world. Well, vision is not the only sensory matter, but the thalamus is very involved in sensory issues. So you see here the sensory and associative thalamus, and many of my students know that I drill them all the time. All what input goes into the ventral anterior lobe of the thalamus, and you residents should be glad that I'm not asking you in front of everybody. The answer is all limbic system information drives into the VA nucleus of the thalamus. I want to point something out here that's rather interesting. Here is input from VA and thalamus, and look, it's over here, connected with the cerebellum. Well, now, how does limbic information get coordinated with cerebellar information? Cerebellar information is about coordinating movement, balance, things like that. Well, soon, I think we are going to see that cerebellar influence in the diencephalon and in the limbic system is much more influential than we've ever understood. The cerebellum is only 10% by volume of the brain, but it is 50% of the brain's neurons. And the brain, in evolution, didn't use 50% of its neurons to completely ignore the emotive system. And we're going to know more and more about this. Now, in this sensory associative connection, you can see some of the intricate pathways here that are involved, driving down into the motor neurons themselves, the sensory systems. You see the sensory motor loop, and you see these subcortical regions in the middle that are largely driven by dopamine. How much influence in these subcortical regions, how much influence occurs in any given region, has much to do with the amount of dopamine in the area at the time. So neurotransmitters, and we are going to come back to neurotransmitters in a minute, they're very important, but interestingly, Dr. Goodkin, Dr. Moore, and I recently had a discussion in which Dr. Moore said that Mark George, a friend of ours from Medical University of South Carolina, made a comment in Norman's journal. I don't know about neurotransmitters anymore. I really think it's about electrical transmission. And that is a fascinating comment, because neurotransmitters are still critically important to our understanding of clinical psychiatry, no question about it. But I think that his comment about understanding the electrical coordination of the brain is going to become increasingly important. And that's where it is, things like cerebellar influence in the sensory thalamus is going to become important for us to understand. These are areas that are not well known at the current time. So what is better known is we can now develop these connectomes. This is a telencephalic connectome, and we're looking at the human frontal lobe. And there's some interesting things to note here. First of all, we think of the brain as a visual organ. But from the frontal lobe, vision is no more important than auditory, nor what I refer to frequently as idiovisceromotor influence, or information processing down from the frontal lobe through the insula into the gut and the body. But then something that the frontal lobe definitely does all the time is compare memory information and take that memory information as a comparator, moving it into the cortex for relative valuation, because that is very important. It also receives tremendous amount of associative information from the cortex in general, the parietal lobe in particular. So this is what the telencephalon looks like in terms of its complicated wiring diagram. And these connectomes are interesting because no behavioral neurology talk is really complete without mentioning Phineas Gage. And of course, in 1848 in Cavendish, New Hampshire, Mr. Gage unfortunately used the tamping rod you see there in his hand to tamp down some black powder, which then ignited and shot that tamping rod and the injury you see below him through his orbit, okay, causing him to have essentially the first prefrontal lobotomy. Now, this was a left-sided lobotomy with a little bit of right-sided damage. But interestingly, the graphic computational capacity now is such that we can use his skull case, which is on display in New Hampshire, and we can actually graph the neural damage that occurred, and we can actually even go in and graph the connectome with the damage that you obviously see in the large missing area of neurons there. And so this is one way that we can begin to visualize brain structures in such a way that we can see how things work and how things don't work. We can go a little further than that. We can use proteins, and we can use these same kinds of connectomics by looking at proteins and looking at human heritage from our African genesis, and we can actually map these proteins so that not only can you know what your heritage is genetically, you can also find out how that plays into the evolution of the brain series that you're using. That's why I said it's an individually personalized brain. And we'll talk about the individuation of the human brain a little more at a later slide. So all of these mechanisms are available. Great graphics are available to us these days. This is mouse retina. And I put this up particularly because it plays into another mechanism we can use for understanding the nature of brain connectors. Fractal mathematics, and if you look at these carefully, you'll see how closely they resemble fractal mathematics patterns. Now, why is this important? Well, if you look at these fractals, in particular, you see how complicated they become right in this mid-region. Well, why is that important? Another thing that is really not well understood currently is what we refer to as the hidden layer. So we know a lot about sensory input. We know a lot about receptors, and we know a lot about motor output. But when you go back and think about that thalamus slide with its complicated wiring diagram that I put up, talked about cerebellum, that hidden layer of how all these intercoordinated interconnections act, that is still very unclear to us. And as we develop better computer models, better mathematical models, we'll be able to look at this hidden layer more and more specifically. And as we understand it, we'll be able to see the actual electrical patterns that exist. So eventually, these telencephalic and whole brain connectomes will allow us to understand much more about various pathological states. And that's important because we already understand now quite a bit about brain networking. For instance, the default mode network. This is essentially what happens if you're lying still on your bed with your eyes closed and sort of actively not thinking anything. So you're in kind of a daydream state. And in this situation, default mode as it's referred to, the midline regions are largely activated, okay, and other networking functions are deactivated. The central executive is largely Brodmann's areas 9 and 10, and those areas of the prefrontal cortex that I showed you and referred to as executive function regions. Once you snap to attention, those regions rapidly come online. And then the orbital frontal regions and the associative regions that are involved with the frontal lobes begin to develop the question of saliency. Not only are you concentrating and attending to things, but you're deciding what is important and what isn't important. You're valuing things, and that has much to do with how the saliency network works. So being able to map these networks very specifically will give us a tremendous amount of insight into things that we care about in psychology and psychiatry. Areas like, what is the nature of reward? Well, first of all, we know that dopamine is very heavily involved in reward, and the blue lines here, especially the thick blue lines, are dopaminergic lines. And we know that dopamine is involved, amongst other things, with the nucleus accumbens. Okay? There's lots of information from the frontal cortex and the orbitofrontal regions reaching into, and you'll notice here, ventral striatum. Many of you recall that I've pointed out the ventral striatum is nucleus accumbens, subthalamic nucleus, and ventral tegmental area. So these areas are heavily involved with dopamine, heavily involved in the nature of pain, pleasure mechanisms, and reward. They have a lot of limbic input. And down here, they go, you'll notice right in the middle, they go to VP. Why that? What is it? Well, it's the ventral pallidum. The pallidum is the globus pallidus interna and externa. And its ventral 1/3 is the emotive region of the globus pallidus. And that ventral 1/3 is probably the most primitive emotional processing center. So back in the day, the Cambrian explosion, when it was bugs crawling around on rocks, not much emotive processing, maybe none, not much real consciousness. But as they began to progress, even as they got to worm level and beyond that, nematodes, then they got to the place where this ventral pallidum began to coordinate sensory motor loops in such a way that there was actually a valuation of what that was. And that then evolved into higher animals, moved on eventually to both sea and land critters that had more capacity to put emotive influence in these sensory motor loops and make sense of them. And as that process developed, the importance of neurotransmitters arose, very important to understanding consciousness. Now, the two big brain systems are glutamate and GABA. They're huge. Okay? And they didn't start off first. Instead, those three little systems that some of you have heard a psychiatrist or two refer to occasionally, norepinephrine, serotonin, and dopamine, we occasionally talk about those. Those three systems act as trophic systems in development. So glutamine and GABA systems don't know where to go until norepinephrine or serotonin or dopamine beacons tell them where to go in the developing brain. So these systems are actually quite small compared to glutamine and gamma. They're very small, okay? But they are hugely important in development. And they also essentially, norepinephrine basically acts as a channel changer. Serotonin basically acts as if it were the modulator that connected your remote to your TV in a simple metaphor. Dopamine is largely the volume switch. So dopamine is very important because I show a diagram of limbic systems here that make the point about how understanding these neurobiological functions can enhance psychiatry. You'll notice here the habenula, which is at the back of the thalamus, pushing down to the interpeduncular nucleus. And right here is the area of the central gray. In the central gray, endorphins and enkephalins exist. And the stria thalami medullaris goes from the area of the septum and the nucleus accumbens, basal forebrain nuclei, goes to the habenula to help control the habenula in a dopamine loop that then presses on the interpeduncular nuclei in such a way as to modulate the nature of endorphins and enkephalins in the periaqueductal gray. This means the things that we're used to using for affect and for psychosis can also be used to modulate pain symptoms, especially chronic pain symptoms. So this information begins to help us understand how these things actually work and what they can mean clinically. Now, an interesting thing is, it's only recently, really, that we understood that the ventral tegmental area, pressing dopaminergic information into the nucleus accumbens, which would be excitatory information, also has an excitatory pathway, glutaminergic pathway, descending into the region of the ventral tegmental area as well. Well, now glutamine, that's actually the universal neurotransmitter. So we should have universal excitatory neurotransmitter. So we should have this pathway where dopamine is exciting and glutamine is exciting, and it should be a feed-forward loop. It should just go completely haywire, out of control. But it doesn't. Why? Because there are these GABA interneurons in the system. GABA, the universal inhibitory neurotransmitter, actually slows this system down in a modulatory kind of way so that response to pain, pleasure, reward can be effectively modulated. Well, those are interesting ideas. You see I also want to bring up the role of the ventral pallidum. You see how closely related it is there to the nucleus accumbens. So the ventral pallidum, very ancient evolutionarily, but the nucleus accumbens, a modern variant on its theme. All of it involved with driving emotional controls. The amygdala, of course, is also involved with emotional control, and not at the cortical association area. The amygdala processes the world looking for fear-threat information. It compares that fear-threat information with historical memory. It decides how important it is, and there's some important ideas about this. Up here, again, not on this yellow line, but right in this region, again is the head of the caudate. And in fact, the head of the caudate could be thought of as actually the tail of the amygdala. Well, the head of the caudate has that bidirectional connection to the frontal lobe. The amygdala turns fear-threat on whenever something unknown comes up. And then there is some processing at the frontal lobe level. What am I going to do about this? And then another interesting item in the brain comes into play, another one of the basal forebrain nuclei, the septal nucleus, which turns the amygdala off if it is overreactive to an alien stimulus of some sort. All of this connects into the nucleus accumbens, where pain, pleasure, reward, dopaminergic materializes. And all of this is connected by way of the bed nucleus of the stria terminalis in such a way that we get very quick sensory motor feedback in terms of fear-threat information. Yeah? The diencephalic part is almost completely unconscious. So the material in the limbic system ascends into the anterior cingulate cortex where it turns into motive drive, and it is not conscious. It's a subcortical associative pathway set. But once experienced, it's going to be transferred into the hippocampus for later comparator value, and from the hippocampus, it appears that what happens is the information goes to level 1 of the cortex at the associational levels and is therefore stored as long-term memory, which can then be reaccessed later. And actually, I very much appreciate your asking a question, because I'm going to come to a slide in a minute called the Mary problem, and it has a lot to do with that. How do we know what something is? That is an important question in consciousness. But I am on the unconscious level of the diencephalon right now, and we're looking at it. You see the amygdala. See its close association with the hippocampus, less connected than we might think given their close connections, the ventral tegmental. And if you'll look here, you specifically see the insula. This is the portion of the brain, large portion of the medial temporal lobe that is involved with idiovisceromotor activities, gut responses to thoughts and emotions. And then you see, of course, the cingulate gyrus again, that area which is in its anterior portion, pushing motive drive forward into frontal lobe. Here's a look at the septal area. Now, the septal area is turning the amygdala off when the amygdala is over-controlling, causing too much fear and panic. But it also is interestingly connected to the acetylcholinergic nucleus basalis of Maynard, famously associated with Alzheimer's disease. And in this nucleus in particular, acetylcholine plays this interesting gating role, deciding how much information should be allowed at threshold in various regions. So the septum needs to be active enough to turn the amygdala off, but not so active as to cause the amygdala to be ignored. We need to understand fear threat. and we need to do something with it. The brain itself is a liquid, solid interface computer. It's important to remember how vascular the brain is. And it's very important because vascular injury causes tremendous damage in these network functions. Here we are looking at the basic structure of how sensory relay comes in, is processed at various levels, is integrated with emotional and motor program selection, is dealt with by the cerebellum, and then ultimately results in motor relay. In addition to that, I bring this slide just for Dr. McCarley because every time I put it up, she calls that, oh yeah, a Hendrick slide. This shows how integrally related and how neat these networks are. And obviously, nobody needs to try and memorize this. These networks, though, are controlled, we used to think by neurotransmitters, we used to think by neuronal DNA, but in fact, the astrocytes that are largely involved in building the blood-brain barriers can determine whether or not a given synaptic activity can occur because the astrocyte can actually disallow calcium to be in the synapse. If it does, neurotransmitter cannot be released, and therefore that synaptic connection is currently offline. So these are many different ways of looking at the complexity of the brain. Here is a DTI, diffuse tensor image, of an owl monkey brain. Now, this owl monkey eventually was sacrificed, so we got tremendously good resolution on this. This is a human brain, and you can see there's less great resolution. How do we go about these connectomes? Well, at first, we get a brain, which eliminates most humans. We get a piece of it. We cut it very thin, make an image, reconstruct it, and then make visual analysis. And we go down further and further and map out how many connectors there are in very small areas. So we zoom in to get these specific connections, and having enumerated them, we then can develop, by computerizing the numbers, we can develop these networks, and the larger the circle, the greater the number of synaptic inputs into a nodal area of the brain. Well, to get here, we start off by basically looking at centrality and what hubs in the brain are most connected. From there, we build rich club graphics. And rich club graphics, as you can see here, rich club graphics can be in any variety of ways. This idea comes from the idea that people meet in social clubs to run communities. Well, the brain kind of meets in its own social club. And some social clubs are going to be thick and dense, others less so. But still, whatever the nature of these rich clubs are, they're going to be very influential, either by white matter tracting or by particular bipolar neuron associations or by mirror neurons. From there, then, we develop a rich club map with its connectors. And from that rich club map, we move on to get a decomposed connectome of the overall brain, and from there we can overlay a graphic look at the overall brain. And if you look at this particular brain, there are a couple of interesting things. Look at these large number of midline modal connectors. This would be a brain demonstrating default mode network to a certain degree. Also out in the amygdala regions, there's quite a bit of connector. Well, how did we go about getting this level of graphic? We use these eigenvectors. And eigenvectors, if you think about this as a neuronal microtubule or even a neuronal axon or dendrite, there's Brownian motion of the molecules in this region. And these molecules are bouncing back and forth. They are separating from one another, but they also align at times, and they also have a certain cohesion sticking together to the average position, because biology requires that to be the case. What you can do with water molecules in this way is you can apply a strong enough magnetic current for a long enough time, and the longer you apply, the stronger the current, the more you get resolution in these DTI images. This has gotten so sophisticated that we can look at the neuronal connection networks themselves. And if you look at this and you think about it for a minute, just look at those blue neurons. Let's say those blue neurons were growing towards norepinephrine trophic signal in development. The orange ones were growing towards a serotonergic trophic signal. This is the way the brain lays down the pattern of the complexity of its neuronal structure that it holds. When we then get enough information that we can actually look at the brain overall, we see some interesting things. Look, very influential rich clubs down here. What's there? Oh yeah, the amygdala are there. Need to watch the world for fear threat. And what's here? Anterior cingulate cortex. Also very important to take all that associational information at a subcortical, as Jay points out, unconscious level, drive it into the frontal lobe for modulation into consciousness. Genetics has a lot to do with this too, and we shouldn't forget that. This is a look at a MET variant. This is a network in an autistic child. Our resident just presented us with Rich Club networking paper on autistic children. I thought it was very interesting. I thought of this when he showed it to me. This is interesting because if you get this genetic methionine variant, okay, substituting for valine in the MAOA gene, these children then have a dyssynchrony or a relative disconnection syndrome that allows them not to have a very effective daydreaming network. In sociopaths, this kind of incoherence can occur in such a way that they can be fearless in their nature. And this same variation of MAO seems to be responsible for that. Another way that this information can be helpful is we can superimpose it from the interior brain that I've been showing you to look at the exterior brain by using tractography. And then as we return to the interior of the brain, we see some very specific tractograms here. And there are several of them. First of all, I mentioned the arcuate fasciculus, the big green one here. This is going basically from the anterior temporal lobe tip through Wernicke's area, through the angular gyrus over to Broca's. This big red one is corticospinal tract. This is superior longitudinal fasciculus. And this one in particular is the uncinate fasciculus. The uncinate fasciculus, when it has poor coherence from frontal to temporal lobe in sociopaths, seems to be intrinsically involved with the behavioral patterns that sociopaths and psychopaths demonstrate that other people don't. So this idea of coherence, what do I mean? Well, when you talk about resolution, coherence, we're really talking about the electrical activity of the brain. I come to the discussion that Dr. Moore brought to our attention. And in this electrical activity, we see ascending influence going from the midline reticular formation, which in every person in this room, which in every individual in this world, is specific only to that individual. It's very hard to study because it doesn't have these typical patterns. Every one of us has developed our own way of coordinating information flow in the reticulum. And from that information, there is a press into the interlaminar nuclei of the thalamus. From those interlaminar nuclei of the thalamus, then, there is a pulsation that runs at about 40 hertz, and that information basically powers the cortex. And you see here that the septal nucleus, responsible for turning off the amygdala if overactive, nucleus basalis of Maynard, are also directly involved with these electrical translations. And it makes sense. One is turning off fear. The other is gating information flow. Okay. Oh, good. Hit the wrong button. I wondered what happened. All right. So here's that 40 hertz pulsing pattern coming out of subcortical brain pacemakers to drive the cortex. Now, if we were at the level of electrical information flow in the hidden layer, we could understand much of what is going on in the unconscious levels of the diencephalon. We're not there. We're on the surface of the brain. We can look at superficial EEG. We know these thalamic reticular nuclei are driving the information But all we can do right now is map the surface information. Nonetheless, that leads to fascinating information. Our brains can choose the next action we're going to make as long as 10 seconds ago. That's a long time, a very long time. In consciousness, there is this disconnect. We have a sense that we're in control of our consciousness, but in fact, our brain is running these underlying patterns of understanding all the time, well in advance of things that we actually make conscious decisions about, leading us to the Mary problem. How do we know that Mary is Mary? Or in this particular instance, how do we know that red is red? Because these sensory motor feedback loops are so sophisticated. They push associational material, they make historical comparators, they make multiple comparators. All of this takes time. And so any given perception we have comes to us after multiple reafferent loops, comparators, and reconsiderations of what a given stimulus is. So this leads us to Edelman's idea, Tonini and Edelman's idea, of what is consciousness itself. Well, those bugs crawling around in the Cambrian explosion didn't have much consciousness. Humans have a tremendous amount of consciousness. I ask you, is your dog conscious? Your cat? How about if you have a pet snake? Is it conscious? Well, it is, sort of. But I think the degree of consciousness between a bug, a snake, a dog, is tremendously different, because the more complex the cortical material is, the more consciousness evolves, develops, and drives specific behavioral features of life. So the neocortex then is the associational area for memory. It's inhibiting limbic information, unconscious information. This is all controlling the brainstem and motor activity. So what are the minimal physiologic correlates for consciousness? Well, now, I talked about snakes and dogs, but I didn't mention chickens. Are chickens conscious? Like, for instance, how about Mike? Here's Mike, the headless chicken. Mike got his head chopped off by the farmer that intended to have him for dinner, but something happened. Mike didn't quite die. This was in the 1930s. Mike had a brainstem. Mike had a little portion of his superior temporal sulcus, so he had a little bit of auditory capacity. And he lived like this for 18 months, walking around the chicken yard, okay? No head, just little bits. Well, was this being conscious? Not very. On the other hand, Dr. Griffith kind of surprised me today. I was dug into the computer. He walked in behind me, and I about jumped out of my shoes. So, you know, he activated my auditory superior temporal sulcus in my brainstem. So how much more conscious am I than Mike? I guess it depends on how much I'm into my computer at a moment. This chicken actually grew from 2.5 pounds to 8 pounds in this headless condition. So it brings us to this fascinating question. What is consciousness? Well, quantum mechanics tells us we have to have a massive number of digital components to be able to explain or even begin to theorize an explanation of consciousness. But if you look at these microtubule associations in individual neurons, and if you think about the number of microtubules that must exist in all of those 100 billion neurons and all of those synaptic connections and all of the complex DNA neurotransmitter interactions that occur, if we look at these microtubules and you notice these proteinaceous connectors, the little Z's here, between each microtubule, well, you can imagine that at any given point in time, at any given place in a neuron, in your brain, these things can be in either one format or another, giving them the ability to be digitized. They're either 0 or 1 at a given moment. And that number of microtubule connectors and that number of potential either on or off status is a large enough digital number that probably quantum mechanics could begin to explain things about neurotransmission, about behavior, but we have to have these graphical ways of looking at the brain, these mathematical ways. We have to understand the genetics, the neurotransmitters, the networks. And what does all this have to do with psychiatry? Well, our job is to understand what an integrated brain is. I hope I've made some progress on telling you how complex that job is in this story. And then once we know how integrated it is, we want to know if there's a coherent mind that exists in that integrated brain. If so, we bring empathy to try and modulate these things in such a way that we can make a positive difference. And with that, I thank you for your time. Questions? Yes, Dr. Good.
John, you mentioned the ability of astrocytes to modulate calcium in substances that's affecting neurotransmitters. Is one thing anything about balance between neurotrophic factors and pro-inflammatory cytokines and chemokines?
You know, Dr. Brown, when I asked Dr. Brown that question, he said, well, yes, that's a whole new lecture.